Hey everyone, and welcome to another deep dive into the world of medical microbiology.
It's great to be here.
Glad to have you. Today, we're tackling a group of bacteria that might not always steal the spotlight, but they're definitely worth our attention.
Oh, absolutely. They can be quite the sneaky troublemakers.
Exactly. So we're talking about non-beta-hemolytic streptococci. And to guide our exploration today, we'll be referencing Principles and Practice of Clinical Bacteriology by Gillespie and Hockey. It's like our trusty roadmap through this microbial jungle.
A classic. It's always on my desk.
Same here. Now, for those who might be thinking Streptococci, isn't that just strep throat? Well, get ready for a plot twist because the world of Streptococci is much more diverse than you might think.
Oh, yeah. It's a whole extended family with different branches, and each one has its own unique quirks.
Okay, so let's start by untangling this family tree. What exactly sets these non-beta-hemolytic Streptococci apart from their more infamous relatives?
Well, the first thing we microbiologists do is see how they behave on a blood agar plate. It's like their first impression.
Right. You can tell a lot about bacteria by how they interact with blood cells.
Exactly. So some Streptococci, the beta-hemolytic or pyogenic group, they completely break down red blood cells, leaving a clear zone around their colonies. It's a pretty dramatic entrance.
Like a bacterial rock star trashing the hotel room.
Exactly. But our non-beta-hemolytic Streptococci, they're a bit more subtle. Some, like the alpha-hemolytic or viridin Streptococci, cause partial breakdown, leaving a greenish discoloration.
More of a coffee shop jazz vibe.
I like that analogy. And then we have the gamma-hemolytic group. They don't cause any visible change at all on blood agar. They're the quiet ones observing from the corner.
So how do we tell these quiet ones apart? It seems like just looking at them isn't enough.
You're right. It gets a bit more complex. Traditional methods like serological grouping, which uses antibodies to detect specific antigens, or observing their hemolysis patterns, they have their limitations. It's not always a clear-cut identification.
So we need to dig a little deeper.
Exactly. We've shifted towards analyzing their DNA, their genetic blueprint techniques like DNA hybridization and 16S ribosomal DNA sequencing give us a much more accurate picture of their family tree.
So we're going beyond outward appearances and peering into their genetic code.
Precisely. It's like comparing their family photo albums to see who's really related to whom.
I love that analogy. So what have we learned from peeking into their family albums? What does their family tree look like?
Well, it turns out there are 6 major branches in the streptococcal family tree. For today's deep dive, we'll focus on the branches most relevant to human health: the anginosus, mitis, salivarius, mutans, and bovis groups, along with the nutritionally variant Streptococci.
Okay, already sounds like a diverse crew. And I'm guessing each group has its own preferred hangout spots in the human body.
You got it. Many of them are part of our normal flora, peacefully coexisting with us. The anginosus group, for instance, can be found in the oral cavity gastrointestinal tract, and even the genital tract.
So they're quite adaptable.
They are. And they're known for causing abscesses, those painful pus-filled pockets of infection.
Abscesses. That doesn't sound fun. What about the other groups? Where do they like to set up shop?
Well, the mitis group is a bit of a mixed bag. Some, like Streptococcus pneumonia, are major pathogens, causing everything from pneumonia to meningitis. Others are common residents of the mouth and can cause endocarditis, a serious infection of the heart valves.
Endocarditis. We'll have to say that for another deep dive. But let's stick with these non-beta-hemolytic streptococci for now. We've got the anginosus group causing abscesses, the mitis group with its mixed reputation. What about the salivarius group? The name kind of gives it away.
You're on the right track. They're primarily found in the mouth, especially on the tongue. They have a bit of a sweet tooth, producing a sticky substance called leaven from sugar.
Wait, so they're contributing to that dreaded plaque my dentist is always warning me about.
You guessed it. And then we have the mutants group, the notorious cavity-causing crew. They're like tiny sugar-fueled factories producing acids that eat away at tooth enamel.
Okay, I'm starting to see a pattern here. These non-beta-hemolytic streptococci might seem inconspicuous, but they can definitely cause their fair share of trouble. We've got the abscess-forming anginosus group, the mixed bag mitis group, the plaque-producing salivarius group, and the cavity-causing mutants group. What about the bovis group and those nutritionally variant streptococci? Where do they fit into all of this?
The bovis group is interesting. They were initially found in cattle, but they can also cause infections in humans, including endocarditis. And some strains are even linked to colon cancer, although the exact connection is still being investigated.
Colon cancer? Now that's intriguing. And what about the nutritionally variant streptococci? What makes them so picky?
These are the divas of the streptococcal world. They're a bit trickier to grow in the lab because they need specific nutrients to thrive. They're often found in the throat urogenital tract and intestines, and like the others, they can also cause endocarditis and other infections.
Okay, so we've got this diverse cast of characters, each with their preferred hangout spots in the human body. But here's the million-dollar question. If they're often part of our normal flora, why do they sometimes turn against us and cause infections?
That's the fascinating part. They're what we call opportunistic pathogens. They usually only cause problems when there's a change in the host's susceptibility or their local environment.
So it's like they're waiting for the perfect opportunity to strike.
Exactly. A weakened immune system, an injury, or even a disruption in the normal balance of bacteria can give them the chance to cause mischief. And a key event in many of these infections is something called bacteremia, the presence of bacteria in the bloodstream.
So something as simple as a cut or a dental procedure could potentially lead to bacteria in the bloodstream.
It's possible, especially if you have any underlying health conditions. But don't worry, a healthy immune system usually takes care of these transient invaders.
That's reassuring. But going back to those more serious infections like endocarditis, what makes these seemingly harmless bacteria capable of causing such a major illness?
Well, it's a multi-step process, but essentially these bacteria have developed some clever strategies for invasion. They use adhesins, which are like molecular grappling hooks, to latch onto damaged heart tissue.
So they're not just floating around aimlessly, they're actively seeking out vulnerable spots.
Precisely. Once they have hatched, they establish themselves within a forming vegetation, which is like a fortress built from platelets, fibrin, and bacteria on the heart valve.
So they're building their own stronghold right on the heart valve itself.
That's a great analogy. And from there, they can grow and multiply, causing further damage and potentially leading to serious complications.
That's a fascinating, albeit slightly terrifying glimpse into the world of these opportunistic pathogens. But I'm sure our listeners are wondering, are all non-beta-hemolytic streptococci equally adept at causing endocarditis?
That's a great question, and the answer is no. Some species like those in the mitis group are more commonly implicated than others. Their interaction with platelets seems to play a significant role in the severity of the infection.
You mentioned earlier that some streptococci can induce platelet aggregation. How does that contribute to the development of those vegetations?
Imagine these bacteria releasing signals that cause platelets to clump together. This clumping, along with the bacteria themselves, forms the building blocks of those vegetations on the heart valve.
So they're not just passive passengers. They're actively manipulating the host's own cells to create a more hospitable environment for themselves.
Exactly. And to add another layer of complexity, they've evolved specific mechanisms to survive within those heart valve vegetations, like enzymes that repair damage caused by the host's immune response.
It seems like these bacteria are masters of adaptation and resilience. This is all incredibly insightful. But let's shift gears to the clinical side of things. If we suspect someone has a non-beta-hemolytic streptococcal infection, what are the telltale signs? What should clinicians be looking for?
That's where the diagnostic challenge comes in. The symptoms can be quite subtle, especially in subacute endocarditis.
So it's not always a dramatic presentation like with some other infections.
Not always. Patients might initially experience fever malaise, a general feeling of being unwell, and perhaps a heart murmur. But these symptoms could also be indicative of many other conditions.
So it's not always clear-cut.
Exactly. And to make things even more complicated, other signs like emboli blood clots that travel from the heart to other parts of the body, cardiac failure, or an enlarged spleen might develop later on.
So it's a bit of a puzzle requiring careful observation and a good understanding of the patient's history.
Precisely. That's why laboratory diagnosis plays such a critical role in confirming the presence of these bacteria and guiding appropriate treatment.
Speaking of diagnosis, how do we actually identify these bacteria in the lab? What tests are involved?
Well, the first step is always careful specimen collection. We need to minimize contamination whether we're working with blood pus or tissue samples.
So proper technique is crucial from the very beginning.
Absolutely. Then we typically culture the sample on specific media, providing the right environment for the bacteria to grow. We might use blood agar selective media for certain groups, or media tailored for those picky eaters like the nutritionally variant Streptococcus.
So we're trying to create the perfect conditions for these bacteria to reveal their true identities.
Exactly. And once we have some growth, we confirm that we're dealing with gram-positive, catalase-negative coccia arranged in chains or pairs. That narrows down the possibilities considerably.
So those initial screening tests help us rule out other types of bacteria.
Precisely. From there, we use biochemical tests to differentiate between various streptococcal species. And in some cases, we might even use molecular techniques like 16S rRNA gene sequencing for a definitive identification.
It sounds like a multi-step process requiring a good deal of expertise. Now, the source material mentions some intriguing shifts in infection patterns over the years. Can you tell us more about those trends?
You're right. The epidemiological landscape is constantly changing. Data from England and Wales shows a decline in infections caused by the sanguis group, while the mitis and anginosis groups are on the rise.
So the dominant players are shifting.
That's correct. And this is likely influenced by various factors, including changes in healthcare practices, antibiotic usage, and even the aging population.
So it's a complex interplay of factors.
Absolutely. And it highlights the importance of ongoing surveillance to track these trends and adjust our prevention and treatment strategies accordingly.
That's a perfect segue to our next topic, prevention and control.
Ah.
Given the increasing antibiotic resistance we're seeing with these bacteria, how can we effectively protect individuals from these infections?
Well, one of the most fundamental strategies, especially for infections originating in the mouth, is good oral hygiene. This might sound simple, but it can make a world of difference.
So brushing, flossing, and regular dental checkups are still our frontline defense.
Exactly. And for patients with predisposing conditions like damaged heart valves, antibiotic prophylaxis before certain dental procedures is often recommended.
So we're trying to prevent those transient bacteremias that could lead to a more serious infection.
Precisely. But the key here is careful risk assessment and appropriate antibiotic selection. We want to avoid overuse and unnecessary exposure, which can further fuel antibiotic resistance.
It's all about striking that delicate balance between prevention and responsible antibiotic stewardship.
Absolutely. And speaking of treatment, that's where things are getting increasingly challenging with these non-beta-hemolytic streptococci.
Yeah, treatment is definitely where things get tricky, especially with this rise of antibiotic resistance. But, before we dive into that, I think it's worth revisiting those abscesses we talked about earlier. They're not just a messy inconvenience. They can actually be quite complex.
Okay, abscesses, right? Those are those painful, pus-filled pockets of infection, right? What makes the anginosis group so good at forming them? Do they have some kind of secret weapon? Well, they do have a few tricks up their sleeve. Remember those adhesins we talked about, those molecular grappling hooks? Well, they use those to attach to various tissues and build up a fortress.
So they're really good at establishing themselves and holding their ground.
Exactly. And to make matters more interesting, those abscesses are often polymicrobial.
Polymicrobial, you mean they invite other bacteria to the party?
Precisely. Studies have shown that co-infection with certain anaerobic bacteria can actually make those abscesses more severe. It's like they're forming alliances to boost their pathogenic power.
Oh, it's not just a solo act. They're teaming up with other microbes to wreak havoc.
That's a great way to put it. And it seems that those anaerobic partners might even be helping to suppress the host's immune response, making it easier for the Streptococci to thrive.
Wow. That's sneaky. It's like they're creating a whole underground network of infection. But we mentioned that the anginosis group is associated with abscesses in various parts of the body. Is there any connection between the specific species and where that abscess pops up?
You're thinking like a true microbiologist. And the answer is yes. Research suggests that Streptococcus intermedius seems to have a preference for the central nervous system and liver, while Streptococcus constellatus is often found in lung abscesses, and Streptococcus anginosus tends to stick to the gastrointestinal and genitourinary tracts as well as soft tissues.
So it's almost like each species has its own travel itinerary.
Exactly. Although we're still trying to understand the specifics of why they choose certain destinations. But remember that human-specific cytolysine we discussed earlier, intermetallysin.
Oh, right. The one produced by Streptococcus intermedius that can damage host cells.
That's the one. It might be playing a role in why S. intermedius has a penchant for those deep-seated abscesses.
That makes a lot of sense. Now, we know that immunocompromised individuals are more vulnerable to infections in general. What role do non-beta-hemolytic streptococci play in these patients?
That's an important point. With advances in medicine, we have more people living with weakened immune systems due to organ transplants, cancer treatments, and other factors. And unfortunately, they're highly susceptible to infections, including those caused by our non-beta-hemolytic friends.
So these bacteria that might cause a minor inconvenience in a healthy person could pose a serious threat to someone with a compromised immune system.
Exactly. We're seeing a rise in these infections, causing conditions like septicemia, acute respiratory distress syndrome, and pneumonia.
That's concerning. Are there any specific species that are particularly problematic in this population?
Streptococcus oralis, Streptococcus mitis, and Streptococcus sanguis seem to be the usual suspects. They can sneak into the bloodstream from the oral cavity or the gastrointestinal tract, especially when those mucosal barriers are compromised.
So it's like a combination of a weakened defense system and a breach in the walls that allows these bacteria to launch a full-scale invasion.
That's a great analogy. And to add another layer of complexity, some of these streptococcal strains are becoming resistant to commonly used antibiotics, making treatment even more challenging.
So it's a double whammy of increased susceptibility and limited treatment options.
Unfortunately, yes.
Yeah.
That's why prevention is absolutely crucial in this population. Good oral hygiene, careful monitoring for any signs of infection, and judicious use of prophylactic antibiotics are all key.
So it's all about minimizing risk and acting swiftly if an infection does occur.
Absolutely. Now, before we move on, I think we should briefly discuss another important player in the streptococcal world, dental caries.
Ah, yes, those pesky cavities. I think we all know someone who blames their sweet tooth for their dental woes.
Indeed. But while sugary treats certainly play a role, the real culprits are the bacteria that thrive on those sugars and produce acids that attack tooth enamel.
And I'm guessing the mutants group of Streptococci are the masterminds behind this tooth-decaying operation.
You got it. They have several characteristics that make them highly effective at causing caries. They're acidogenic, meaning they produce acids from sugars. They're also aciduric, meaning they can survive and even thrive in those acidic environments they create. And to top it off, they produce those sticky extracellular polysaccharides, especially from sucrose, which help them cling to teeth and form plaque.
So they're essentially building a sugary, acidic fortress on our teeth, slowly dissolving the enamel away.
That's a great way to visualize it. And those frequent sugary snacks and drinks provide them with the fuel they need to keep the destruction going.
This all highlights the importance of good oral hygiene and limiting those sugary temptations to keep those cavity-causing bacteria in check.
Absolutely. Regular brushing, flossing, and professional cleanings are essential for disrupting plaque formation and removing those acid-producing bacteria. And let's not forget fluoride. It helps strengthen tooth enamel and make it more resistant to those acid attacks.
Okay, so we've covered the major players in the non-beta-hemolytic streptococcal world, and there are various strategies for causing trouble. Now, I know we can't diagnose an infection based on symptoms alone. Laboratory testing is crucial for confirming the presence of these bacteria and guiding treatment decisions. What are the key steps involved in the laboratory diagnosis of these infections?
You're absolutely right. Lab diagnosis is crucial for pinpointing the culprit and choosing the right weapons to fight back. The first step is getting a good specimen, whether it's blood pus from an abscess, a wound swab, or even a biopsy.
So it's all about collecting the right intel from the scene of the crime.
Exactly. And it's important to avoid contamination. We don't want any innocent bystanders throwing off our investigation. For example, if we're sampling an oral abscess, we'd use a syringe to aspirate the pus rather than a swab, which could pick up those friendly neighborhood bacteria that are just passing through.
That makes sense. We want to make sure we're catching the real perpetrators. So once we have a clean specimen, what happens next?
The next step is to grow those bacteria in the lab, giving them the right environment and nutrients to thrive. We use media that contains ingredients like meat extract, peptone blood, or serum to cater to their specific needs.
So we're essentially setting up a 5-star hotel for these bacteria, pampering them so they'll reveal their true identities.
Precisely. We might use general-purpose media like blood agar or more specialized media that select for specific groups like TYC agar for mutans Streptococci. It's like setting out a buffet that only certain guests will find appetizing.
I love that analogy.
Yeah.
So we're not just growing any bacteria, we're creating conditions that favor the growth of the suspects we're most interested in.
Exactly. And we also have to consider the incubation conditions. Remember, these are facultative anaerobes, meaning they can grow with or without oxygen. We might need to adjust the atmosphere to mimic the conditions in the human body.
So we're creating a home away from home for these bacteria, making them feel comfortable enough to multiply and reveal themselves.
That's a great way to put it. And once we have some growth, the real detective work begins. We start by observing the colonies, their size, color, and whether they cause hemolysis on blood agar. These initial clues help us narrow down the possibilities.
So it's like we're gathering evidence from the crime scene, looking for any distinctive features that might point to the culprit.
Exactly. We then run a series of tests to confirm that we're dealing with Streptococci, such as the Gram stain and the catalase test.
Right, those are the classic microbiology tests. I remember learning about those in school.
They're the foundation of bacterial identification. But sometimes those initial tests aren't enough to definitively identify the species. That's when we turn to biochemical testing, where we look at their ability to ferment different sugars, produce certain enzymes, or grow in the presence of specific inhibitors.
So it's like we're analyzing their metabolic fingerprints.
Precisely. Each species has a unique biochemical profile, which helps us pinpoint their identity.
And I imagine technology has made this identification process much faster and easier.
You're absolutely right. We now have commercial identification kits that incorporate a panel of biochemical tests and a database for rapid species identification. It's like having a fingerprint database for bacteria.
That's incredible. It must be a huge time saver, especially when you're dealing with a potentially serious infection.
Absolutely. But it's important to remember that even with these advanced kits, there are times when we still need those traditional methods. And in some cases, we might even turn to molecular techniques like 16S RNA gene sequencing for the ultimate confirmation.
So it's a combination of classic techniques, cutting-edge technology, and good old-fashioned microbiological know-how. Now let's talk about the elephant in the room, antibiotic resistance. We've already touched on this, but what are the implications of increasing resistance among non-beta-hemolytic Streptococci?
That's a critical issue that's keeping us all on our toes. The rising resistance to antibiotics like penicillin macrolides and even some of the newer drugs is making treatment much more difficult.
So the weapons we once relied on are becoming less effective.
Unfortunately, yes. And a big part of this is due to the overuse and misuse of antibiotics, which has driven the evolution of resistant strains. It's like we've been inadvertently training our enemies.
So it's a classic example of survival of the fittest.
Precisely. When we expose bacteria to antibiotics, we create a selective pressure that favors those with resistance mechanisms. It's an ongoing evolutionary arms race.
I see what you mean. It's like we're constantly trying to stay one step ahead of these microscopic masterminds. But how exactly do these bacteria develop resistance? What are their strategies for evading the effects of antibiotics?
They have a whole arsenal of tricks. One common mechanism is enzymatic inactivation, where they produce enzymes that break down or modify the antibiotic, rendering it harmless. It's like they have a team of ninjas disarming our weapons before they can strike.
That's a great analogy. So they're essentially neutralizing the threat before it can even reach them.
Exactly. Another strategy is target modification, where they change the structure of the antibiotic's target site, making it harder for the drug to bind and do its job. It's like they're changing the locks on their door so our keys no longer fit.
So they're constantly evolving to outsmart our strategies.
They're incredibly adaptable. Some bacteria can even reduce the permeability of their cell membranes, making it harder for the antibiotic to enter. Or they might develop efflux pumps, which are like tiny pumps that actively expel the antibiotic from the cell. It's like they have a security system that detects intruders and kicks them right back out.
It sounds like we're up against a formidable opponent.
We definitely are. And this is why antibiotic stewardship, using antibiotics responsibly and only when necessary, is absolutely crucial. We need to preserve the effectiveness of these life-saving drugs for future generations.
Absolutely. Now, given this challenging landscape of antibiotic resistance, how are we currently managing non-beta-hemolytic streptococcal infections? What are our treatment options?
Well, as we've discussed, the first step is accurate identification. We need to know exactly which species we're dealing with to choose the most effective antibiotic. And susceptibility testing is essential to ensure that the chosen antibiotic can actually kill the specific strain causing the infection.
So it's all about tailoring the treatment to the individual bacteria, just like personalized medicine for humans.
That's a great way to put it. We also need to consider the site of infection and ensure that the antibiotic reaches a high enough concentration to be effective.
So it's not just about choosing the right weapon, it's also about making sure it reaches the battlefield in sufficient force.
Exactly. And the duration of treatment is crucial as well. We need to administer the antibiotic for a long enough period to completely eliminate the infection and prevent a relapse.
So no cutting corners or stopping the medication as soon as you start feeling better.
Absolutely not. That's a recipe for treatment failure and could even contribute to antibiotic resistance.
I see what you mean. So we need to make sure we completely eradicate the enemy and prevent them from regrouping.
Exactly. And for some infections like endocarditis, we might use combination antibiotic therapy to enhance efficacy and reduce the risk of resistance development. It's like using a multi-pronged attack to overwhelm the enemy.
So we're pulling out all the stops to fight these infections.
We definitely are. And as we mentioned earlier, in cases of endocarditis, surgery might be necessary to replace damaged heart valves and completely remove the infected tissue.
So it's not always a simple fix with antibiotics alone.
Indeed. Endocarditis is a serious condition that often requires a multidisciplinary approach involving cardiologists, infectious disease specialists, and surgeons.
It sounds like treating these infections is a team effort requiring a combination of clinical expertise, microbiological knowledge, and careful monitoring.
You're absolutely right. And it's important to stay up to date on the latest treatment guidelines and emerging resistance patterns to ensure we're providing the best possible care.
This has been an incredible deep dive into the world of non-beta-hemolytic streptococci. But before we wrap up, I have a challenge for our listeners. Are you ready for some exam-style questions to test your knowledge?
Bring it on. I'm sure our listeners are up for the challenge.
All right, question number 1. Which of the following streptococcal species is most commonly associated with brain abscesses? Is it A, Streptococcus aeruginosus, B, Streptococcus constellatus, C, Streptococcus intermedius, or D, Streptococcus mutans? Think carefully about what we discussed regarding the different species and their preferred anatomical sites. Take your time, and we'll reveal the answer and explanation after a short pause.
All right, time's up. The correct answer is C, Streptococcus intermedius. Remember, this species has a knack for causing those deep-seated abscesses, particularly in the central nervous system and liver. And don't forget its secret weapon, intermetallicin, that human-specific cytolysine that can wreak havoc on host cells.
Great explanation. Ready for question number 2? Here it is. Which of the following properties is not characteristic of the mutans group of Streptococci? Is it A, acidogenicity, B, aciduricity, C, production of extracellular polysaccharides, or D, beta-hemolysis on blood agar? Think back to our discussion about the mischievous mutants group and their role in dental caries. Which of these characteristics doesn't quite fit?
The answer is D, beta-hemolysis on blood agar. Remember, the mutants group are non-beta-hemolytic, meaning they don't cause complete lysis of red blood cells on blood agar. They might cause alpha hemolysis, greenish discoloration, or gamma hemolysis, no change but not beta hemolysis. They're all about those acids, those sticky polysaccharides, and their ability to thrive in acidic environments.
Excellent. Now for question number 3. Which of the following is a key event in the development of most non-beta-hemolytic streptococcal infections? Is it A, bacteremia, B, viral co-infection, C, ingestion of contaminated food, or D, exposure to environmental toxins? Think about how these bacteria, often part of our normal flora, transition from harmless residents to opportunistic pathogens. What's the critical step that allows them to spread and cause trouble?
The answer is A, bacteremia. Remember, bacteremia is the presence of bacteria in the bloodstream. And for many of these non-beta-hemolytic streptococci, gaining access to the bloodstream is their ticket to ride, allowing them to travel to different parts of the body and cause infections. This can happen through various means like dental procedures, injuries, or even something as simple as vigorous toothbrushing.
Right. It's a reminder that even those everyday activities can have unintended consequences.
Yeah.
Okay. Ready for question number 4? Here it is. Which of the following antibiotics is typically recommended for the treatment of penicillin-sensitive viridans streptococcal endocarditis? Is it A, vancomycin, B, erythromycin, C, benzylpenicillin, or D, ciprofloxacin? Think back to our discussion about treatment options and the importance of choosing the right antibiotic for this specific bacteria and infection.
The answer is C, benzylpenicillin. It's the gold standard for treating penicillin-sensitive viridans, streptococcal endocarditis, usually combined with gentamicin for added firepower. Vancomycin is a good backup option for penicillin-resistant cases or for patients with penicillin allergies. Erythromycin and ciprofloxacin, however, are not effective against most streptococcal species.
Excellent. And now for our final question. Which of the following is the most important preventative measure for minimizing the risk of developing infective endocarditis in individuals with predisposing cardiac conditions? Is it A, avoiding contact with sick individuals, B, maintaining good oral hygiene, C, wearing a mask in crowded places, or D, getting vaccinated against influenza? Think about where many of these non-beta-hemolytic streptococci reside and how they gain access to the bloodstream. What's the simplest yet most effective way to reduce the risk of infection?
The answer is B, maintaining good oral hygiene. Remember, many of these bacteria call our mouths home. By practicing good oral hygiene, including regular brushing, flossing, and dental checkups, we can keep their numbers in check and reduce the risk of them sneaking into the bloodstream. While the other options are important for overall health, they don't specifically target the root cause of endocarditis in individuals with predisposing cardiac conditions.
That was a fantastic review. Hopefully our listeners are feeling like true streptococcal experts now. But remember, this is just a glimpse into their complex world. There's always more to learn, and research is constantly uncovering new insights.
You're absolutely right. It's a fascinating and ever-evolving field, and we're just scratching the surface.
Speaking of scratching the surface, let's delve a little deeper into the fascinating world of streptococcal taxonomy next. We'll explore the tools and techniques that microbiologists use to classify these diverse bacteria and uncover their evolutionary secrets. Stay tuned. Welcome back, everyone. Before the break, we were about to step into the world of streptococcal taxonomy.
Yeah, it's a fascinating area. You know, it's like solving a microbial mystery.
Okay, I'm all ears.
Yeah.
So how do we actually classify these diverse streptococci? What are the key characteristics that taxonomists are looking for?
Well, it's kind of like building a family tree, you know? Traditionally, we relied on phenotypic characteristics, those observable traits like their appearance, their growth patterns, and their biochemical reactions. Remember how we talked about those alpha hemolytic, beta hemolytic, and gamma hemolytic distinctions on blood agar?
Right, those were our first clues about their different personalities.
Exactly. And biochemical tests, like the ones we use for diagnosis, can further differentiate them based on their metabolic capabilities. What they eat, what they produce, how they respond to different environments, it all adds to the picture.
So we're essentially profiling their lifestyles and habits.
Precisely. But as technology has advanced, we've incorporated genotypic analysis, which means looking at their DNA, their genetic blueprint. And this has really revolutionized our understanding of bacterial relationships.
So we're going beyond those outward appearances and peering into their genetic code.
Exactly. Techniques like DNA hybridization and 16S ribosomal DNA sequencing have become invaluable tools for bacterial taxonomy.
Can you explain how those techniques work? I'm a bit rusty on my molecular biology.
Sure. DNA hybridization is like comparing blueprints. We measure the degree of genetic similarity between two organisms. The more similar their DNA, the closer their family ties.
So it's a measure of genetic kinship.
Exactly. 16S ribosomal DNA sequencing focuses on a specific gene that's present in all bacteria and evolves relatively slowly. By comparing the sequences of this gene from different bacteria, we can construct phylogenetic trees, like family trees, that show their evolutionary relationships.
So it's like tracing their ancestry through their genes.
That's a great way to put it. And this approach has been incredibly useful for clarifying the classification of Streptococci and even identifying new species.
Okay, I'm starting to see how powerful these genetic tools are.
Yeah.
So how has this revolutionized our understanding of the genus Streptococcus?
Well, for starters, it's helped us move beyond those simple classifications based solely on hemolysis. Remember those viridin Streptococci, the ones that cause the greenish discoloration on blood agar?
Right, we talked about them earlier.
Well, genetic analysis has shown that they're actually a very diverse group with many different species, each with its own unique characteristics and pathogenic potential.
So it's like we've zoomed in on what appeared to be a single family and discovered a whole neighborhood of distinct individuals.
Exactly. And this has huge implications for clinical practice. We can no longer assume that all viridin Streptococci will behave the same way or respond to the same treatments.
So accurate identification is more important than ever.
Absolutely. We need to know exactly which species we're dealing with to make informed decisions about treatment and prevention, and this applies to other streptococcal groups as well. Genotypic analysis has helped us refine our understanding of the Agenesis, mitis, salivarius mutans, and bogus groups, giving us a much clearer picture of their roles in human health and disease.
It sounds like taxonomy is a constantly evolving field, always refining and updating as we gain new knowledge.
You're absolutely right. As new species are discovered and our understanding of their relationships deepens, we adjust our classifications accordingly.
This really highlights the dynamic nature of science. It's not about fixed answers, but about constantly questioning, exploring, and refining our understanding of the world.
Well said. And this collaborative spirit is essential for tackling the challenges posed by these non-beta-hemolytic Streptococci, especially in the face of increasing antibiotic resistance.
This deep dive has been an incredible journey. We've explored the diverse world of these often overlooked bacteria, from their unique characteristics to their pathogenic strategies, the challenges of diagnosis and treatment, and even the intricate field of bacterial taxonomy.
It's been a whirlwind tour, but I think we've successfully illuminated some of the complexities and clinical significance of these fascinating microbes.
I completely agree. I think the key takeaway here is that while these non-beta-hemolytic Streptococci might seem like quiet members of our microbial community, They're capable of causing a wide range of infections, from minor annoyances like cavities to life-threatening conditions like endocarditis.
Absolutely. And their growing resistance to antibiotics adds another layer of complexity to the challenge.
It's a reminder that we're in a constant dance with these microbes. We need to stay vigilant, adaptable, and innovative to stay one step ahead.
Well said. I think this deep dive has equipped our listeners with the knowledge and insights they need to approach these non-beta hemolytic streptococci with a newfound understanding and respect.
I couldn't agree more. And to our listeners, thank you for joining us on this exploration. Keep those curious minds engaged and never stop exploring the amazing world of microbes. Until next time.